In this video, we will study about transcription. Transcription is formation of RNA. RNA is ribose nucleic acid. Formation of RNA over DNA template is called transcription. Now, this is the DNA strand. Now, DNA is deoxyribose nucleic acid. Its one strand is in the 5' to 3' direction, and its second strand is in 5' to 3' direction in this direction. Now, out of the two strands, one is called as non-template or coding strand. Second is called as DNA template or antisense strand. Now, this strand of the DNA, which is running in 3' to 5' direction, is called as DNA template. Now, this is the strand on which RNA will be produced. DNA is double-stranded, RNA is single-stranded. If this strand of DNA is in 3' to 5' direction, now RNA transcript will be produced from 5' to 3' direction. Supposing this is the sequence on the DNA, GCA, GCC, TTC. So opposite to G, C will be produced, opposite to C, G will be produced, and opposite to A, U will be produced. Now in case of RNA, thymine is not there. Thymine is replaced by uracil. So uracil is represented by U. So opposite to A, U will be produced. So similarly, opposite to GC, then C, opposite to CG, opposite to TA. So in this way, bases will be produced. So this is the single strand RNA which is being produced from this strand of DNA. So this is called as RNA transcript sequence. Whatever the sequence will be present in the 3' to 5' direction, complementary, and opposite to that will be produced in the RNA transcript. Now, what are the enzymes which are required for the transcription? Now, enzymes required for transcription are RNA polymerase. Now, we have two kinds of organisms, prokaryotes and eukaryotes. In prokaryotes, bacteria and blue-green are included, and in eukaryotes, animal cell, plant cell are included. Now, in prokaryotes, only one RNA polymerase help in formation of RNA, but in eukaryotes, we need to have RNA polymerases which are of three types. RNA can be of three types: messenger RNA, transfer RNA, and ribosomal RNA. So depending upon which RNA is to be produced, RNA polymerases are going to help. RNA polymerases 1 are for pre-ribosomal or pre-ribosomal RNA. 2 is for pre-messenger or pre-messenger RNA. 3 is for pre-transfer or pre-transfer RNA. 5 sRNA, small nucleolar RNA. As we know, what is RNA? It is the ribose nucleic acid. Now, RNA polymerase in prokaryotes mean bacteria and blue-green algae, where well-defined nucleus is not present. Now, what is the structure of RNA polymerase in prokaryotes? It is made up of 5 kind of polypeptide chains. Now, these are 5 type of polypeptide chains. We can see 2 alpha, 1 beta. Now, this is beta prime. So, this is the sigma factor of polymerase. Now, this helps in recognition of the start signal or promoter region of the DNA from where transcription has to start from the DNA strand. This is called as transcription initiation site. Now, this is 6 polypeptide chain model of the RNA polymerase. In this case, we can additionally see omega factor. Rest is the same, 2 alpha, beta, beta prime, sigma, and this is the omega factor. Now, RNA polymerase has core enzyme and sigma factor. So, this is the core enzyme and this is the sigma factor. Now, when they may be dissociated like this and they may be associated like this. Now, this is alpha, beta, beta prime, omega. Now, this is the strand of DNA from 5' to 3' direction. This is 3' to 5' direction. So, here is the promoter region on the DNA to which this RNA polymerase is going to recognize and bind to it. Now, this is the RNA polymerase which is 5' polypeptide chain model. This is the sigma factor. Now this is the binding site, and this is the initiation site from where RNA transcription will start. Now in case of eukaryotes, some other factors are required for initiation of transcription. A number of proteins are required, and these are called as transcription factors. TF2D, TF3A, TF3C, TBP, TAF. And some are enhancers which increase the rate of transcription, that is formation of RNA. And some are also called as silencers, which repress or reduce the rate of transcription. And some of the protein factors, they are required for the chain elongation, TF2F, TF2S. And when RNA transcription has to be stopped, then terminator region is required. Termination factor is Rho, which help in termination of the RNA formation. Now again, this is the DNA strand, this is the promoter region where RNA polymerase binds, and this is the initiation site, and this is the gene in case of eukaryotes. It's not simple, it is called as split gene. Now split gene has exonic region and intronic region. So this is exon, intron, exon, intron, exon, right? So whenever exons they will be joined, introns has to be removed. So that is called as process gene. And this is the point where RNA transcription formation is terminated. So this is called as transcription terminator region. So promoter region is present on the DNA, and this is that region on the DNA where RNA polymerase will bind and start the transcription. So this is about transcription. Thank you for watching my video. If you like my video, please like, share and subscribe. In second video, we will discuss exact mechanism of transcription, how it occurs.